working. Okay, thank you. Now we can start recording. All the embarrassing things have been said. Okay. Yeah, I just wanted to close with 2 minutes because Andrea wants me to burn through a little time to give people opportunity. We're up to 53 now, so that's excellent. And of course, there are hundreds here in the room. You can't see us all, but it's wall-to-wall packed here. So just some comments on why I think pangenomics is so important. Rob Pike, a famous programmer who worked at Bell and is responsible for one of the Unicodes, the 8-bit Unicodes that is able to encode 1 million+ characters, mentioned that really data dominates. And what he meant by that is that if you organize data in the right structure, that then the algorithms can be relatively stupid. So this goes back to the even earlier quotes of this sort, that if you get your data structure right, the rest follows, not organically, as those of you who code will know, it's still a hassle, but you need to get the data structure right. And the pangenome is the right graphic data structure for life on Earth. It's kind of embarrassing to think retrospectively about how all of us bought into the concept of a reference genome. 70% of the public effort of the human genome, which was started in 1990 and kind of sort of finished in 2003, 70% of that genome was from one individual. Can you guess the ethnicity of that individual? Actually, an African-American. Did you guess that? So from Buffalo. So that's the famous RP11. I'd love to meet RP11, but 70% is from that one individual. And then there were another 19 individuals who contributed the other 30%. So it's not quite as bad, perhaps, as the Celera effort, where it was all one genome. Can you guess who that was? Craig Venter, of course. I don't know if you said that, but yeah. So this notion of a reference genome is a complete and utter kludge that it should have been obvious from day one, probably was obvious from day one, but it made life easy. So it's, just it's been a 23-year hack now. So we're at the 20th anniversary of the original release here with the pangenome now in 2023. And now I think we can get the data structure right by using pangenome graphs as the data structure, not only for humans, but for life in general. And data dominates. That's my motto. That's why I don't know how to code. I do know how to clean up data. So Andrea, yours.
Thank you, Rob, for your kind introduction. It's a pleasure to introduce the first speaker, Mr. Zigui Bao. Zigui, you can start to share your screen, please. Perfect. Okay. Zigui received his bachelor degree in life science in 2016 at Nanshan University. Then he worked as a bioinformatics engineer in several companies in China, developing genomic pipelines for plants and animal populations. Before his PhD, Zigui was part of the Dr. Sang Wenhang group at Agricultural Genomics Institute at Shenzhen, where he led projects on tetrasomic inheritance on autotetraploid potato and on using pangenome graphs to capture missing heritability in tomato. Now, Zigui is pursuing his PhD at the Max Planck Institute for Biology in Germany, where he's exploring the high diversity region in Arabidopsis thaliana and studying transposable elements with pangenome graphs. He's also working to improve the pangenome graph builder pipeline. Today, Zigui will show us the mysteries that plant genomes hide and how we can face them by applying pangenomics approaches. Zigui, thank you for being here virtually. Please, you can start.
Okay, Thank you, Andrea, for your kind introduction, and thank you for inviting me for this amazing conference. I hope I can be at Memphis in some day. And today I want to show you with the complex genome rearrangement we found in the pangenome graph. And this background is from how the AI thought pangenome graph should be. Actually, in reality, it's more crazy than this one. Today I want to show you. And one foundation problem of the genetics is how do we understand how the relationship between the genotype and the phenotype. And in the plant, the diversity is quite high. When we're looking back at the phenotype, they may have a different leaf in abdomens, and they may have a different fruit shape or color in tomato and potato. And in the maize, all the amazing maize is caused by the transposon, a lot of cases. So the diversity is very high. And another approach from the genotype, we didn't have, in the past, we focused on the SNP and the small variant from indel. And now we shift the focus from the structure variation. So it's more maybe the 5 major type, but some was very complex. It's more than two structure variation. So the relationship between the genotype and the phenotype, how can we find this? So the basic idea is from when we have a phenotyping from the different individual, and then we genotyping them by the sequence-based methods. And within short reads, we can find SNP or indel, and then we can do the linkages mapping. Like the first figure is we using the cross, and we have a mapping population. We do the QTL analysis, and all we use the natural population. So, but the problem was when we do the GWAS, we found maybe we didn't have enough the significant signal, or we only can find a major allele. Why this is a problem? So this problem is start from the human. The human started GWAS in 2006, and they found this problem we call the missing heritability. So take human height as an example. The heritability estimated by the twin study is only about 18%. But when you using all the variants, all the significant SNP from the GWAS study, it only could be explained 16 variants. So this gap we call the missing heritability. So this problem could be caused by the incomplete lineage disequilibrium between the variants. Also could be the genetic heterogeneity. What do you mean the genetic heterogeneity? When you have a different mutation, but they will cause the same phenotype, this will decrease the power of the GWAS. So we have the allelic or the locals. Also, it could be the small effect in allele or the environment interaction. So in 2010, the Jian Yang, he developed a tool and using all the SNP, not only the significant SNP, to estimate the heritability of height. This could be increased into the 45%, but we still have gap. So the idea problem was what we can do expect for the statistical model is we want to include in all variants or more possible the variant in one individual or in the population. So we started from the tomato first. So in tomato project, we using the expression data as a phenotype. We comparison 2 datasets, one dataset from the linear reference, this is the blue one, and another dataset from the graph. We genotype structuration using the graph. Then we found when you're looking back, sorry, well, I can show a pointer. Okay, so when you're looking the structuration from the graph, it can have increased much more heritability. And when you including all the variant to explain heritability, it increased more than 10%. And we also partitioned this heritability in the joint estimation, and we found the structuration explained more than half gene in our expression data set. So we think about the structuration is more likely to be the causal variant, and it more have a much larger effect than SNP. We using the giraffe for mapping, and we using short reads for the genotyping. But we found some problem when we do the tomato graph project. So the problem is always the mapping. So the challenge of the variant calling, when even you have a graph, you still have two mapping reads to graph. The mapping will fail when you have a repeat. It could be the subtype, different subtype where they are very similar, and the read is very short. You cannot distinguish the variation or the copy number variation between the different similar or transposon. And also could be the large scan generation, like inversion, is much longer than your read length. So this is the very hard problem. When people do the short read, we have a problem. When you do the long read, even have a problem. Look in this, in some region, even the long read, it could be mapping. So you using the echelon or something more longer. This is the limitation of the mapping or the genotyping. Another problem from the this variant calling is the VCF limitation. We using VCF to store variation, but actually some variation is very complex. We cannot using the standard VCF to store them, like this loci in the tomato. We have this loci is caused by the different tomato flavor, and when you think this is have a deletion, and then this have a maybe have a duplication, also have a point mutation. And when you want to including, or this is loci in one VCF, this is impossible. So this is a challenge of the variant, how we can do the ideal way. Actually, for the ideal way approach for the variant calling, we use in assembly. So now we have the T2D genome of human, and many species is to produce the T2D genome. Every base pair we can capture in one single genome, and in large scale sequence project, every species could be a model species. We didn't need to choose one specific species as model. Now also, we didn't need one reference. We can using every individual as the reference. This is the pangenome. So the pangenome, this idea started very early in the 2005 using the bacteria. They're using the multiple genome. They define the core genome and dispersal genome. And for the pan, it's just map. You get all the genome, all the multiple. You can find the transposon or the variation in one set. But how can we represent this variation? So this is how the graph emerged. The variation graph. It developed by Eric. And this is from the ES from his paper. And this we use in the node, age, and path to represent the graph. This graph is very similar to the subway map. So using this graph from the different genome, we can explore the variation space within one species or maybe cross species. So how can we build a pangenome graph? I think the workshop already introduced a lot, but I want to summarize this pangenome universe. The first part approach is we using the VG or the variation graph toolkit using the VCF to building a graph to the mapping. And then the second effort is from the cactus. So it developed for a whole genome alignment, and using the guide tree to do the alignment. Then this alignment itself could be a graph. Another approach from the mini graph, it choose one reference, and then mapping the second one, the third one, they just merge the similar and to introduce the new sequence or in the graph. They didn't have any path, they just a combination of the all structuration. So this both effort have a limitation. So they come up with an idea to combine these two projects in together. This is called the mini graph cactus. So using the mini graph first, and we then using the cactus to do the base level alignment. Then you have a new alignment. It didn't need a guide tree, and it be more requirement of the reference base, but it's still reference based methods. So it's in the middle, it's part of the reference base, but part of the reference free. Another way is from all the totally reference free is it's PGB, pangenome graph builder. We do the auto alignment. And we using the alignment graph to transfer to the variation graph. Then we smooth the graph and to finally get consensus. Another way is not do the whole genome alignment, but it's only explore some region. So this approach could pangenome research toolkit. So this idea comes from the assembly graph. So using the minimizer in the sequence, when you give a one query region, they can get a retriever all the homolog sequence in the pangenome, then using this sequence, the minimizer, the common minimizer, to building a graph, and then using the different window, different kimmer to explore this graph. How can this infer different haplotype or different variation? So we have a lot of graph, but why we need, how do we choose, how do we, what the disadvantage of the advantage? How can we use the first when we compare with the PGB and the mini graph? This is an example from our paper from the tomato graph pangenome. We found one specific structuration. This structuration is highly correlated with the gene expression because it truncates 5' region. So it's 2.6 kilobase deletion in this. And when we revisiting this graph or using all the assembly we have, we found this have a more complex one than we thought. So this have a 3 haplotype. The first haplotype is basically totally similar with the reference. This first one is the reference. And this second haplotype is deletion in the gene region. Also 1 deletion in the intron. So in this region is 8.6 kilobase deletion. And also it's a very large deletion in total intron. So this is the whole LTR transposon. So it's more than 12 kilobase. So we found this will be more complex than we thought. But this 2 complex structuration didn't include in the genotyping approach in the giraffe because we have a 1 specific step to merge to the duplication of the variation structuration because the VCF limitation. But when we look in the graph from the PGB, so this is the deletion from the gene region, and this is whole the transposon deletion, and this is a part of deletion. It only remains 1 solo LTR. But when we're looking back for the mini graph, this graph is totally different. And when the intron region, because the transposon, the mismapping, so this is more fragment mapping, and we thought they have a lot of variation. But when we're looking back at assembly, they didn't have so much variation, or at least it's much simple. It could be simplified in the 3 major haplotype. So that's why we want to do comparison between the different pipeline. So we're using the 4 different species, the public plant genome data set, one from the Abdominal sienna to the crazy maze. They have like the genome size range from the 100 megabase to the 2000 megabase, and the transposon from the 60% to 18%. So, but for the benchmark, we need the ground truth. Actually, we not unlike human, we didn't have the genome important, so we didn't have the ground truth to do the benchmark. But what we want to do, just using the cross validation first. So from the tomato, we using the variant from the deep variant as the ground truth, or just the one true set for the comparison, because small variant is more easily to call, and with the high fidelity is more easily to call. And when we compare from the PGB graph and deep variance call set, all the SNP is very similar. It's more than 19%. So the SNP is very perfectly, or at least it didn't have too much problem. And we found in the major 3 group, this group represent the SP is white tomato, and the SLL is the cultivated tomato, and the SLC is the landrace tomato, and the reference is from the cultivated. And because the divergence between the different accession, and this in there have a huge difference. When you look in the precision, it's very low, but when you look in the sensitivity, they are still very high. So what does it mean? It means the graph include all the possible variants, but they are different represent. But because we didn't have a ground truth, we didn't know which one is true, especially for the small in there. So we just found this different disequilibrium, and we found one specific outlier from 2 accession. The accession have a very large inversion. It's from the it have more than 40 megabase. And the first thought we maybe have a misassembly, but when we mapping back the high rates to the reference or to the assembly individual, it look the carriage is very even. So it didn't have a it did have a misassembly, at least for this inversion. And when you mapping again when you mapping the reads to the reference, the coverage is uneven. So this discrete may be the variant from the deep variant. They didn't have it including the more, or maybe the graph, they have a different approach to deal with the variant. So this is only for the small one. We want to know how about structure variation. So we revisit the NSGT1, the example I presented before. So it have a duplication and deletion in one paper. And we also using our high-res genome, using the graph, we found this have a duplication and the deletion, and even some region maybe have a inversion, but we didn't know it's true. But at least it have a good continuance with the previous reported. And this loci, when you're using the mini graph, and we cannot really understand what happened here, and also the graph structure will be slightly different because the mismapping also in the PDB graph also have a small chunk for the mismapping. So another complex case is always we try a lot of approach to explore this complex loci in the tomato. It's in the subtomato region. It called Tomlock C. So it influence the QTL influence a lot of metabolic in the tomato, but we didn't have the truly causal variant, or not only one causal variant. And when you're looking the mapping again, do the alignment between the wild tomato and the reference, and this is have like huge, the much more copy number expansion, and also the divergence between the different expressions. And when you're looking the graph is very fragmented. And another crazy way from the plant is the maize. We know the maize have a lot of transposon. So I try for the very well-known the example from the maize. So this is the one specific loci for the BZ locals. So this is the size between the different 8 accession. It's range from the 15 kilobase to the 150 kilobase. But when you look in the gray one, it's the gene. So the gene is quite conserved between the different accession. But when you look in the intergenetic region, they have a lot of the transposon insertion, even next to transposon insertion. So I try to using the graph to revisiting this structure. But when you try the different like segment or the identity, the graph structure change a lot, and some region even cannot align. So it's unlimited challenge for the graph when this can working in the maze. I think it can work in mostly in all the plants. So that's the reason I started my PhD in the Arabidopsis. This is a small genome. The tomato and the maize graph maybe is too complex first, and we want to know the small genome first. So I using one dataset recently published. We using the complete assembly from 16.6 hi-fi assembly. We explore the centromere diversity, and the first we want to know is from the did Arabidopsis have a large scan variant like translocation or inversion in the whole population. So we using the chromosome community do the auto mapping, and we found the 5 community is very clear. It's just a 5 chromosome. We didn't have the strong signal to indicate the translocation. So we using one specific chromosome to explore the variation in the Arabidopsis. Also, we using the reference-based the structuration calling tool. One is from the exhibition. So this is a deep learning-based method could be calling the complex structuration. We can see all the complex structuration enriched in the pericentromere or centromere region, but some of them like is in the population, they have a population pattern. And also we tried for the sniffer, it's using the population scan, the structuration calling. But when we look in the map, the genotype map, the yellow one indicated the heterozygous score, but as we know, the Arabidopsis is highly inbreeding the line in the white. So this heterozygous, the core is mainly caused by the mapping or the genotype error. is mainly still enriched in the centromere or pericentromere region, but it's still in the autosome region because the divergence from the chromosome, from the reference, sorry. Also, we tried for the reference-free approach from the PGB graph, and the graph looked like it's very well aligned. It didn't have too much. It's not like the tomato or the maize, but when you're looking, they have a specific like inversion in this region, and this also have some specific insertion in one individual. And also this is the coverage depth of nodes, and this have some region have very high the copy number variation. And from the graph structure, totally it's quite linear. It didn't have a circle, but when the centromere region, they are very highly divergent. And the one specific region have a highly have more one specific insertion. But we wonder if this case only in the centromere or pericentromere region. So we're looking back some rapid evolved region. So in our lab, most focus on the NLR gene involved evolution in the population. And the NLR gene class is always in the class, is represented in cluster in the genome, in the plant genome, and they can have a duplication, deletion, a lot of the structuration between the different gene copy number, because it had to compete with the passenger. So this is the rapidly involved region in the epidermal sienna. And also we tried using the first RPW8. This is the another well-known example. So when we're looking back, this is using the PCR-based methods to find this gene is highly variable between the different individual. But when we look in the pangenomics data set, this is much diverse than we thought. This is the reference one, they are perfectly matched. And when we looking other, is like 3 copy or 10 copy or even deletion. So this is mostly like very high divergence between different individuals. And looking the PGB graph, when you choose the reference as path, this is only the red one. And when you're looking, when you try another individual, they can explore the variation space. And we found mini graph have a similar structure, but mini graph didn't have a path. So we cannot know which individual should go through these different nodes. So this is for RPW8. Another crazy case is near the pericentromere region, is RPP1. So it's very similar, but when we're looking back for the exhibition core, it have a high overlap with exhibition core. Also, when we're looking for the genotype, even in this region, it has a lot of positive heterozygous core in the population. So we try to using the gene as a sentinel to retrieve all the homolog sequence to try the different parameter combination to recruit this to retrieve this graph. So this is the graph with one specific parameter. This graph in this region, even in the epidermal sienna, is quite crazy. When you think this is like duplication and the inversion, and also they are very high divergence. So when you're looking back, when you align, they are very high divergence. You cannot have a meaningful alignment. And when we're looking back for the mini graph, they look like every individual is different. They have very highly very complex bubble in this region. And when we looking for the whole chromosome, they're all tangled together. So we're using the different parameters to explore only for one region base. The graph will be in a circle when you try to lower identity and lower the segment. But when you choose the higher identity and the higher segment, they could be more linear, but it still have much more complex bubble. So we didn't know how to explore this. We didn't know the ground truth. So we are trying to know using the simulation, maybe using more complex simulation to explore how can we finally find this truly evolution history of this complex loci. But we have some example in the final, the application, we thought it will be the pangenome-based GWAS. So I have a collaborator with the Yongcai Huang. He have a 200 accession from one specific QTL we want to study, and this is from the PCR-based sequence, and we using this to align with the genome. And from this gene region, they are quite aligned, but this is region from the main, they are quite different. But it's just the upstream of this of gene. So this is still have problem. But when we using the VCF to do the association, this is the 500 base pair of the transposon insertion, and this is the 15 base deletion. And when you compare the trait, they have a highly significant difference. So this is the first effort we want to try for the pangenome base. So take a summary, and we want to high quality graph to explore the variation, but we need to high quality alignment. We have to do this alignment fast enough and precise enough. It have to deal with the transposon activity or highly divergent regions such as the NR cluster. Also, we have to compare with the copy number variation, because in some region they are very high variable. Also in plant, it's more, the large scale circulation isn't real, it's more common in the population. So we want to, the graph model could be represent universally the complex and the simple variation in one single format. But we still in the cycle of building graph. We try to build in the different graph and learn from them, and the rebuilding graph, we finally will be figure out what happened in the evolution history. So in the final step, we want to know is the application. How can we use in this model from the graph to the different model of the GWAS? So I want to thank my supervisor, Dr. Boigo, and all the help from my college. Also, the tomato part is my former supervisor, Sanwen Huang. And also thanks to Eric and Andrea for the help for the graph. And thank you for your attention.
Thank you, Zigui, for your talk. Plants are very complex, but I enjoy every time when you're able to apply this variety of tools to analyze them. Mm-hmm. Are there any questions from the audience? From the chat? No. I would have a question, because plants are very complex because their genome are full of repetitive sequences, transposable elements, crazy ploidy. In your opinion, we have different methods to build pangenome graphs, to make pangenomic analysis. Do you think it's enough, or what is missing? Do you need other kind of tools to better tackle the genome? Which is your opinion?
From my opinion, now the tool is enough for the explore, but for the reality, it's more complex by the alignments. So the alignment is more complicated. It's not because of the graph.
Okay, so we need better aligner, if understood.
Yes.
Of course.
Zigui, a great presentation. Have you noted any horizontal gene transfer in your exploration of graphs?
You mean from the bacteria or from other?
Yeah, bacteria, virus, whatever. I'm just wondering whether that's a special challenge. You mentioned some of the challenges in your summary slide, and I'm just wondering to what extent horizontal transfer is yet another challenge.
Actually, we didn't explore for the bacteria, but we explore from the organelle to the nucleus. So in the abdomen, they have a very large nucleus in the chromosome 2. It's more than 600 kilobase. So this is more common. I think maybe this is more common in the plant, because the plant messenger or plasmid is much more complex than the human, because the human only, I think it's only the 16 kilobase. But for the plant, it's more than 100 kilobase. They have more the transfer from the messenger or the plasmid to the nucleus. We didn't explore for the bacteria, because we more want to focus on the plant itself first.
Okay, thanks. Other questions from the chat? So there's a question in the chat. So how viable is it to build a multi-species pangenome? How do the graphs handle evolutionary distance? How far will they break? So I think the question is, when we build pangenome graphs, the species will be put in it. How much divergency can we represent in your experience?
Yeah, in my experience, actually, it's not depends on the evolution distance, because when you're thinking about the across species, when you align the parameters like human and the chimpanzee, they maybe can quite a good aligned. But when you're thinking about the habitat, even like we try in for the tetraploid, they are very complex. Even in some region, it's more like a cross species. So it depends on how divergent of the region, or it's not only the species.
Okay, makes sense.
Yeah.
Okay, thank you. No, I don't think we have time. If quick.
Just the last one. That's a quick one.
Okay. How many genotypes is the maximum you try to combine into the graph?
I think the maximum we try now is more than 100 abdomen CNR, but we didn't try to push the limit or more because we want to focus the complex one first, not just the scalability. It run it maybe 5 day or 6 day.
Okay, too much. Okay.
Yeah.
Thank you, Ziggy, again for your talk. So we can continue. Let me prepare. Next speaker will be, please, Aaron. We're just a technical problem. Just a few minutes, please. Okay. It's my pleasure to introduce our next speaker, Dr. Arangri. Arang received her bachelor's degree in computer science in 2009, and her master's degree by informatics is 2011 from IFA Women's University. She completed her PhD in 2017 at the Genome Medicine Institute, where she focused on building the first high-quality Korean reference genome. After her postdoctoral training in the Genome Informatics section at the National Institute of Health, Arang is continuing her research there as a staff scientist, focusing on the assembly and evaluation of whole genome from long-read sequencing and other emerging technologies. She's also an active member of the Vertebrate Genome Project, Earth by Genome Project, Human Pangenome Reference Consortium, and Telomere to Telomere Consortium. Today, Arang will show us what it means to have a really complete human genome, and which are the new opportunities that telomere to telomere pangenomes will unlock. Arang, thank you for being physically here. Please, you can start.
Okay. Do you hear me?
Yeah.
Good, good. All right. Thank you, Andrea, for the kind introduction. Thank you, everyone, for being here in person, so I don't have to speak to an empty room. Oh, okay. I can hold the mic. That might be easier for me. Yeah. Okay. Good. Yeah, so in my talk, I'm gonna quickly recap the latest status of the T2T Human Genome Project that we just recently finished with the Y. And then we'll go through some of the challenges that we faced while completing the Y chromosome. And that's actually what I thought will be relevant to the people who are interested in pan-human genomes, because a lot of those are pretty relevant for, and depends on how good you have a good representation of other genomes as well. Okay. All right. Yep. You probably heard that the human genome has been completed from telomere to telomere in April. We released the first T2T human genome, CHM13, and this was built from a combination of PacBio hi-fi reads and Oxford Nanopore ultra-long reads. And it took about 10 years to complete the 92% of the initial human genome for the GRC consortium, but it took 8% to finish the last, it took 20 years to finish the last 8% of the genome. And this is because we needed not only algorithmic development, we needed good technology, sequencing technology to go across these most difficult regions. And what does that mean? This means that we have been ignored those 8% of the genomes, meaning that we have been fooling ourselves, ignoring that region. And what are those regions that have been missed? More than 70% of those regions are localized in the centromeric region or the satellite regions that are attached to those centromeres. And about a quarter of those lie in the segmental duplications, regions which are highly similar or have recently duplicated. And also, most importantly, they do contain genes. They are not just random repeat sequences. And you can also see that we closed a huge amount of gap in the P arm of all the acrocentric chromosomal arms, which includes the ribosomal DNA, which I'm going to touch later on. But there is one caveat in this paper: the Y was not included. This was not because we weren't caring about the Y. It was simply because the CHM13 cell line did not contain the Y chromosome in itself. The cell line itself inherited 2 paternal chromosomes from a sperm, so it did not came with the Y initially. So to complete the Y, we took another cell line from HG002, which has been well benchmarked and studied, and we had a huge amount of sequencing data that we could play around with. And as a result, here is a recap on how the Y chromosome looks like. We have put that on bioRxiv. I see that this is the okay, yeah, sorry about that. Thank you. Yeah, this with the Y chromosome from HGO2, this now completes the CHM13 project and serves, it can be used as an alternative human genome reference containing all 24 chromosomes. Compared to GRCH38, we have no gaps, nothing unplaced, and the quality value is either comparable or exceeds the one from GRCH38. And we did try tremendous amount of efforts to match the Y chromosome quality to the one to CHM13, so we don't get any other biases when doing read mapping based analysis or any other mapping based analysis. And along with the Y chromosome, to serve the purpose as an alternative reference, we built up a browser on UCSC genome browser with and IGV is also kindly hosting CHM13 version 2. You can choose CHM13 version 2 as another reference genome other than your preferred human genome reference, and it comes with the preliminary gene annotation from CAT liftoff. And of course, NCBI does that too. We have since then released additional data sets on our GitHub page. It's there. And also collecting issues collected from the community. And for data sets, we are providing CHM13 reference genomes with the Y PAR region masked for those interested in read mapping directly in the linear reference based approach, and also have a version which replaces the mitochondria for convenience. And also provide several versions of gene annotations. This is because we had to curate a lot of the ampliconic genes on the Y chromosomes. We are releasing the curated RefSeq liftoff version of the annotation, along with the official RefSeq annotations that has been performed by the NCBI and the GenCode version and the ensemble version. Also, for those interested, lifting over existing or available annotations from previous references, we are supporting the lifting over by providing chains that we have heavily curated to match, to be orthogonal with CHM13. And also there are other important data sets. We have a hugely curated amount of repeat data sets published as well. And also a lot of the short and long read alignments are available. Most importantly, we did recall all the 1,000 genomes data set again, de novo this time, and also for the Simon's Genome Diversity Panel. We are in the process of, we did release all the VCF files. You can find them on this GitHub, and we are in the process negotiating with Envil to release the BAMs as well with no egress. And the SGDP copy number estimates, of course, is also available on the genome browser. In addition to that, we lifted over some important data sets that we thought are heavily used in the genomics community, provide them on the CHM13 coordinates. Okay, now I'm gonna switch gear and talk a little bit about from one T2T genome, how we are planning to expand this to the pangenome reference, along with the human pangenome reference consortium. So the telomere to telomere consortium, there are sometimes confusions. What are the differences between the T2T consortium and the HPRC consortium? The goal for the T2T consortium is to produce a gapless genome assembly. Here, for the human purpose, it's a human genome. It could be any genome. And we were majorly focusing on finishing one human genome at a time and trying to build methods that we can more automatically generate T2T complete genomes. On the other, at the HPRC consortium, the goal is to ultimately build a collection of human haplotypes. And with the methods that the T2T consortium is going to build up, and the lessons that we are learned are going to be applied to all the HPRC genomes. So we can have eventually all those haplotypes T2T resolved. And the first draft human pangenome reference has been released. I think a lot of the people sitting in this room were part of it and needs to get credited for that. So to give you just a brief visual idea what the T2T differs from the human genome, the T2T, one human T2T, addresses the completeness biases. So let's say your sample has a variation that's marked in yellow over there. And if your reference is just missing that copy of the yellow arrow part, then all the reads that belong to that region have nowhere else to go, either map to the first copy or don't map at all. But if you have a more complete genome that captures the missing yellow gene copy or the yellow copy, then the reads finally have a home to be mapped on. And the same scenario goes on to the HPRC based or human pangenome based approach. If the sample again has a variation with the more complete reference genomes, now we do have a haplotype that better represents the individual, the sample individual. So we can decide, we can choose not to map our reads to reference A because it maps better to reference B. Okay, so here I'm going to talk about a few challenges that we faced while working on the Y chromosome specifically. And it also covers a lot of the aspects from the entire T2T, the first T2T human genome. So again, going back to the Y chromosome, I chose to talk explicitly about the example of the TSPY ampliconic gene family. It's one of the genes in the Y chromosomes that's coding testis-specific protein. It's only expressed in the testis. And depending on the copy number, it has been known that the expression level differs. So it might have a very important function. And the version in the GRC had a gap in the array. You can see here on top, this is how the TSPY gene annotation looks like here in red. The more thicker, it means it's a protein coding gene. The smaller ones are all the pseudogenes of the TSPY. And you can see that we have captured about 45 TSPY copies here and one TSPY2 copy outside here. And when you look into each copy that's inside the TSPY array, you can see that all of those are in this very repetitive structure. It forms a composite repeat array structure, meaning that there are different types of repeats that form one unit. And you can see that the TSPY is sitting inside this repeat unit. And this entire unit, the composite of repeat, is repeating itself over and over and over. Probably this is why it was so difficult to close this gap initially in the GRC version. And then we noticed that the TSPY2 is located at a different position, right? We first thought, oh, maybe this could have been a misassembly in GRCH38, and we corrected it. But later it turned out through our other companion paper that this was not the case, and there are Y-specific lineages that has the version of TSPY2 upstream of the TSPY array, and that has it downstream of the TSPY array. And we are hypothesizing that there must have been a gene conversion event happening at the end of the inverted repeat 3 that's marked here in black arrow. So the end portion here and here must have been gone through some kind of a gene conversion. But the problem with the annotation this is an actual screenshot of what the NCBI, but you can see from the NCBI comparative genome viewer. Here on top, it's the CHM13. The bottom is the GRCH38 version of the Y. When you look at the same array region, you can see that only partial regions map, right? And a lot of the TSPY gene copies are just annotated as log something, something, something. Yeah, I don't think that this will be really helpful to the community. And this was the biggest motivation why we went on and did release the curated version of the RefSeq annotation. And the other aspect is that when we look at the copy number variation across different individuals, it's highly variable. So imagine that you're trying to build a pangenome out of different Y individuals, and each of the individuals have different copy numbers. How would you like to annotate this? I took the screenshot from our companion paper. Pillay is leading this. It's actually a figure that's in the revised version of the manuscript, so it's not on the bioRxiv version. But what they tried is you can see that they colored each of the TSPY array units into to build a TSPY to cluster them, those that are more similar to each other and those that are more different. You can see that some are clustered more together from this network over here, meaning that they appear more closer to each other. And here I marked the location of the gene TSPY annotation of our T2TY. And you can see that HU38Y contains only a few colored TSPYs in there. And you can see this is ordered by the Y lineage. You can see that only these lineages are having the matching TSPY2 location in the upfront of the array, while all the rest of the lineages have it downstream of the array. So there must have been something happen in between this lineage here. But what I like to highlight here is that using the pangenome representation, it's really helpful to collect and understand more diverse representation of the TSPY copies that we are missing. And you can expand this to other genes, of course, that are not TSPY, that is not TSPY, but are known to be hypervariable across different individuals or across different populations. The second challenge is not only the TSPY, but also we found 2 more composite repeats that harbors these impliconic gene copies in it. And to discover that, it took a huge amount of time and effort to redo the analysis on the repeats because they did not exist before in the HU38. So Savannah and Jessica were leading this tremendous amount of effort to re-annotate and identify all those composite repeats, satellite repeats, transposable elements, such and such. It's laid out in this companion paper of the initial T2T paper. And in there, you can see that they reclassified 62 new repeat entries and submitted them to DFAM, which is used in the repeat masker as a library. And 19 of them were identified as new composites. This excludes the Y, of course. And some of them are located at the acrocentric regions, which were, of course, not known before. And here are the other the other composite repeat arrays that we identified. The other one was the RBMY. You can see that each exon is actually located in between these transposable elements, and also the DAS gene here. But this time, each transposable element is quite like in between or overlapping with the exons here. And they did, Savannah and Jessica redid all the repeat analysis, curated them, and submitted them again to DFAM. So this means that whenever there is a new T2T genome coming up, you might have to go through this entire process again to re-identify missing repeat units that were not identified before. And depending on when you do the repeat analysis, you might get different results, right? The third one is about the palindromes. It was really a big challenge to identify the exact boundaries of each palindromic arms in comparison to GRCH38. But at the end, we did found that the exact inversion breakpoints, there were 2 inversions happened compared to GRCH38. The first one is located inside the P1 to P2 region. And this has been known before that there is a green to red, a green red, and red green inversion. So this palindrome, entire P1 to P3 region shown here, can form such a loop structure because the palindromic arms are so similar to each other. And there can some non-allelic homologous recombination happen in there. And sometimes when it goes wrong, you can completely miss that copy, and this causes very bad effects like azoospermia. You're not going to produce any sperms at all. And we were questioning, is this specific to HG002, or could this be a misassembly? But with the help of Pillai's analysis, we did see that this was not only shown in HG02, this was something that was recurrent among different individuals. But is this Y lineage specific? We don't have a clear answer for that yet, because there were only 1 individual redone for the Y paper, for the Y assembly over there. And the last point I like to touch on is the sequencing biases. So this was a big challenge when we tried to polish and evaluate the heterochromatic region of the Y. You can see more than 50% of the Y chromosome is actually presenting all these beautiful HZ1 to HZ3 copy switches. And this is how an actual HiFi and ONT alignment looks like on the entire Y chromosome. Here I have the satellite annotation. Down here, these GA, TC, AT, GC tracks are showing the microsatellite composition. So when you see a bump up here, it means that this region here is highly enriched with GA, which is known that PacBio HiFi reads are prone and have some issues there. And TC is the reverse complement of GA, so it's gonna have a similar effect. And then there are also these AT enriched regions here. And I'd like to highlight that there are about 5 different HiFi data sets that I realigned to see what's going on. The initial HiFi data set we saw had this drop down here. And the version, this is the same HiFi reads, but rebase called with deep consensus. You can see that the dropout issue is getting better, slightly better than the initial version. But then with the HiFi version obtained from the latest review from PacBio and from WashU, you can see that this bias is almost gone, right? And but surprisingly, when I aligned the same HiFi data, not the same, an older version of the HiFi data from HG003, I could see no biases again. And I was quite puzzled what made this big difference. And it turned out that depending on which library prep kit you are using, you get these results, these different results. So seems like there was a heat induction problem with the version 2 protocol. So any sequencing projects that were produced with the 2.0 pipeline will probably have the similar issue. And for the ONT, you can see that all the two ultra long reads, this one was obtained from the R9, this one from R10. You can see that both are having these jumps and downs, right? But the R10 duplex version has no issues going through it. So there must be some problem with the ultra long reads, ultra long protocols that induces these up and down, or maybe favors some sequence compositions over the others. This is a zoom in into the satellites, so you can clearly see which microsatellite repeat is more associated to one or the other. And then I'm sure that you will ask, so should I use duplex for my future projects and just not rely on anything else? So here, this is a duplex data produced by the HPRC again. And you can see here below another satellite feature that's going up and down, right? This is the HSAT3 region in chromosome 9. It's a huge satellite array in CHM13. And it's going forward, reverse, forward, reverse, forward, reverse. And you can see that at a few regions, when the GA goes up, like for example here, the duplex is completely dropping off its coverage. And this pattern is going on and on and on, along with this microsatellite pattern. And this turned out that Minnow software sees that looks like a block pore. So it kicks out the molecule entirely. So you don't have any sequencing coverage for that. And we discussed this, Sergey Koren discussed this a lot with the Oxford Nanopore, and they are actively working to fix this. A lot of the issues have been already fixed, but I know that they're working to fix some that are remaining. So maybe it's another promising technology. And there are some other remaining assembly challenges. You would probably have seen a lot of those rDNA tangled graph. I thought that all the other talks will have enough graph images, so I decided not to put in one. But it's gonna be the same issue for future projects, where it's difficult to go across these highly repetitive rDNA tangles. The major problem left in there for automated T2T genome assemblies is to find the matching pair of the distal part of the RNA to the proximal part of the RNA. So it's not the centromere this time, but connecting the P arm to the right Q arm is a big challenge. And maybe some additional data can help, like the Hi-C or POR-C data. And we did had some success with the strand-seq data for HGO2, but it's really difficult to scale this up for all genomes, we understand. And then the other challenge is to polish them to the same quality as we had for the T2T-CHM13. There is currently no automated recommended way to polish diploid genomes, but it's an active area, and many people are working on this. And for accomplishing this, we launched a HGO2 Q100 project, where we try to build a benchmark diploid genome assembly of HGO2. So we don't have to rely on the SNP variants that have been provided by the genome in a bottle as a true set when you try to benchmark huge SVs and such, or SV calling methods. Okay, lastly, I'd like to highlight the opportunities that we might get with future T2T genomes. This was a work led by Andrea, who's here. And briefly, if I could explain, the top part here is showing the annotation of CHM13. this large black region consists of the rDNA. And here are all the satellite annotations. This is the centromere, colored in red. And below here are all the assembly alignments against to CHM13. You can see that CHM13, chromosome 13 itself, aligns pretty well because it's the same in the genome. And GRCH38 aligns only up to here because the rest was a gap. And we had HGO2 maternal and paternal draft assemblies available. And you can see that aligns a little bit further down. And each color, by the way, here means which chromosome from this individual segment came from. So everything here in this pinkish color, peach color, means that all these contigs came from chromosome 13. But you can see that going through this region, which has this SST1 gene, we found that it's a pseudo-homologous region. There is a lot of recombination happening in this region. You can see that the beautiful color switches from peach to green, blue, means that it starts from chromosome 13, but then switches to another chromosomal segment. And these PHR, I just make it shortly, PHRs are present on all the acrocentric chromosomal arms. And most importantly, there is one that's in the reverse direction on chromosome 14. And because they are so highly similar to each other, especially the ones on 13, 14, and 21, it's easy to recombine them. And it turns out they are also they exist in the 3D dimension in the proximal location attached to the nucleolus. And which makes it really easy to recombine each other. If it's chromosome 13 and 21, it's going to have no detrimental effects. It's going to be a neutral recombination. But when it happens with chromosome 13, for example, with chromosome 14, then it makes a fusion between these two chromosomes and can produce two different chromosomes and end up having one with 2 centromeres and one without any. And interestingly, this progeny will not have any phenotypic effects, but the phenotypic effects will happen in the child. It can happen, it's called the Robertsonian translocation, and it can cause Down syndromes or any other aneuploidy that you might see. So as a summary, we finished one human genome, T2T, 8% that are now newly accessible. We learned a lot from them. We are working toward building a global pangenome T2T reference set. And with more sequences, we are hoping that we see more variants and hoping to get more clear associations. And there are, of course, challenges ahead. I have shown a few examples of the multi-copy gene annotation problem, repeat annotation problem, identifying palindromes and confirming them, and sequencing biases that affects all, the essential assembly that you are trying to build the genome. And I hope you can also appreciate that the 8% is actually related to this very dynamic region of the human genome. And it turns out it might have more effects than we thought. And it's, of course, happening precisely in the hardest region where it can assemble. And in the future, when the T2T recipes become cheap enough, I hope that we can build our own best reference genome by ourselves and use that to do any other analysis. And this way we can properly measure any somatic variations, I hope. Okay, with that, I'd like to acknowledge a lot of the people from the T2T, HPRC consortium, and especially Adam Philippi, Karen Miga, who is leading the T2T, Sergey Koren, Sergey Nurk, Monica, Savannah, Dylan did the heavy lift for recalling all the 1,000 genome variation calls, and Miko for developing and maintaining Virgo. And of course, Rachel and Monica, no, Katerina for helping me wrapping up the Y project, and also Pillay and Mark and Co for helping with identifying, comparing, allowing to get an early access to their Y chromosome assemblies to confirm our results, cross-validate. Okay, with that, I'll be happy to take any questions.
Thank you, Arank, for your talk. I'm looking forward to build genome complete T2T pangenome graphs, as many species as possible. I have a question, but I would like to give first space for the audience. Any questions from the chat? Okay. So at the moment, to get high quality T2T genome assemblies, we need PacBio hi-fi reads and Oxford nanopore ultra long reads. But we see spoilers that it might be possible, doable just with maybe Oxford nanopore reads, duplex reads. There are people that think that nanopore will dominate other technologies in the future. What do you think about it?
Yeah, I know that it is coming up a lot. I hope that we could in the future just do a simple sequencing recipe. But for now, what I see is that still the Oxford nanopore base pair accuracy, and there are some systematic errors still left in the homopolymer regions, especially, or the microsatellite repeats. So those makes the base pair quality a little bit lower, right? If you don't mind having a less accurate genome, then probably that's the best approach to go with Oxford nanopore alone. Maybe you can obtain some shorter but accurate sequencing technology to polish the genome, but you should be aware that repetitive regions are really difficult to polish with short reads only. So if you're interested in specific genes or repeats, then I guess the answer will be maybe. Yeah, we'll
we have
see.
to choose about base level resolution accuracy or more T2T chromosome resolved.
Yeah, well, more T2T resolved. The T2T relies on ultra long reads. So I think ultra long reads are the essential points. And the question is whether or not you want to switch the hi-fi part to duplex or other shorter but accurate.
Okay, makes sense.
Long.
Thanks.
When you looked at the Y chromosome, did you see any nuclear encoded mitochondrial sequences? So there are probably 5 or 10 of these that are well known that are hard to detect because they're mitochondrial sequence. And I'm just wondering whether the packing of mitochondria in sperm cells has increased the frequency with which you see nummets.
That's a good question. I haven't looked at mitochondrial genomes specifically. Are you meaning about NUMTs, like the nuclear?
They're
Inserted
they're
ones?
just inserted from the mitochondria into the nuclear genome.
Yeah, it's an interesting question. I haven't looked at that specifically. But yeah, it might be not easily detectable because this HEO2 cell line was collected from
Yeah, that's what I was
lymphoblastoid
worried
cell line.
about
Yeah.
actually, because they're, at least in a mouse, one of them appears to be expressed. So it may distort mitochondrial function. Be nice to get them all. They can be as long as 4 kb.
Mhm.
Last question.
I have a question about polishing. I remember in the early T talks that it was mentioned that because of the high quality of the hi-fi, polishing is not needed anymore. Is that not the case anymore, or?
I have a lot to talk about that.
Okay. Okay, I'll catch you up maybe later.
Yeah. Yeah. Well, the short answer is hi-fi still has some issues. And we definitely needed short read polishing for the Y specifically, or the CHM13 to go up to to polish up all those remaining sequencing biases, errors that were caused by those sequencing biases, like the homopolymer lengths differences or other microsatellite lengths differences. Those are a little bit tricky with hi-fi reads only. Yeah, if you really care about high quality, then I'm no. I'm like, no.
Okay, there is a question in the chat, but you can answer later offline. Okay, thank you again for your
Thank
talk.
you.
So we are a bit late, so I propose just 10 minutes for a break and we'll restart.
Everyone. I'm very happy to introduce Dr. Scott Edwards, Alexander Agassiz Professor of Zoology and Curator of Ornithology in the Museum of Comparative Zoology at Harvard University. Dr. Edwards received his BA at Harvard University and his PhD at the University of California. And during his postdoc at the University of Florida, he studied the major histocompatibility complex of birds, an important gene complex for interactions of birds and infectious diseases, pathogens, and mate choice. In the last years, Dr. Edwards was heavily involved in the exploration of avian phylogenomics technologies. Today, Scott will talk about his current research of bird pangenomics. It's a great pleasure to have you here. The floor is all yours, Scott.
Thank you very much. Can everyone hear me okay? Okay, good. Hopefully you can hear me, and it's a real pleasure to be here. I wish I could be at your workshop. I'm sure you're learning lots of useful tools. I thought I'd tell you today about some recent work we've been doing looking at pangenomes of birds. And just see if, how do I, oh, I see, here we go. Okay, yeah, so I don't think I have to explain what pangenomes are. You guys have been talking about them all week. So, but yeah, we'll talk a little about de novo assemblies and pangenome graphs, et cetera. So, you know, the paradigm in ornithology these days is that birds have very small streamlined genomes. And this is some work that I did decades ago with a graduate student. And you can see how birds are, they have very small, very uniform genomes. This is in a comparison with other vertebrates. And this has not really changed in the genomic era. With short read sequencing, we're getting modest genome sizes of about 1 to 1.4 gigabases. And the repetitive fractions tend to be fairly low. And so, for example, here's a summary of some data comparing birds to other vertebrates. You can see the interspersed repeats in this are only 4% of the genome. And so, and the genome size is quite a bit smaller than other amniotes, at least. So that's where we are really. But if you look at the databases, you'll see that, genome sizes are increasing with technology. So you can see, for example, on the right, on the right side of the right graph, you've got contigs and how big the genome size are for those sorts of genomes. Short read contigs, you've got scaffolded short read data, and then in the gray you've got chromosome scale data, and you can see the genome sizes are getting bigger. And this, of course, is averaged across many species. So technology is clearly allowing us to see more of avian genomes. Now, we wanted to look at issues of genome complexity and structural variation in a group of birds that varied in their effective population size. I think most of you are probably aware of effective population size, basically a measure of how much genetic diversity is. And critically, it's a measure of how efficiently natural selection can operate in species with very small effective population sizes, like the island scrub jay you see here on the left. You might expect structural variants to be in high frequency because selection is very difficult. It can't remove them as easily. But in species like the Woodhouse's scrub jay, which you see here is very widespread in the West, has a lot of genetic diversity. We already know this from other studies. We might expect there to be a different distribution of structural variants because selection can act much more efficiently in large populations. And then finally, we've got the Florida scrub jay, which has been studied in detail ecologically for many decades, long-term studies by John Fitzpatrick and others. And so this project was actually conceived during the pandemic. And I was looking for a group of birds that have this characteristic of varying effective population size, and these scrub jays kind of popped up. And so our main goals are really to see how genome structure and fitness effects of structural variants varies across this suite of closely related species that differ in their effective population size. So our genome assembly pipeline was fairly standard now for sort of pangenome era stuff. We did PacBio hi-fi reads on about 45 individuals of the species that you saw, followed by assembly by hi-fi ASM. And then for a few key species and sexes, we improved the assembly with hi-C. And these are just, hi-fi ASM is, as you probably know, a very efficient assembler with long read PacBio data. And this is what some of our data looks like. You can see on the left that the average read length is about 15 KB. You can see that our coverage of each bird is pretty good. This is just one example. You can see the Kamer distributions with homozygous regions, heterozygous regions, and of course, low frequency Kamers, which are likely sequencing errors. These are all outputs from the hi-fi ASM assembly. And then finally, GC content on the right, fairly typical for birds, although we're not quite sure what that jump is at about 50%. Maybe those are gene-rich regions. So our typical assembly looks something like this. This is without hi-C data. We typically get about 1,000 contigs per bird. And as you probably know, Hi-Fi ASM generates not only a primary assembly, but also 2 haplotype assemblies. Each of the haplotype assemblies are about 1,200 to 1,500 contigs. A lot of contigs for sure, but way, way, way less than we would get with a short read assembly genome. And you can see that it's really nice having these phased haplotypes. These haplotypes, of course, are not chromosome scale, but we can really look at patterns of variation on single haplotypes for many genes and gene families, as I'll show you in a bit. Now, with the high C data, you can see we get a big improvement. Here you see the statistics for 3 high C assemblers. There's the primary assembly in red there, and then you can see the SALSA2 and the YAS high C assemblers. YAS stands for yet another high C scaffold or something like that. Anyway, you can see it really made a big improvement in our scaffold N50. And so that's very exciting. And we did choose one bird kind of as a reference, if you will, mostly from the point of view of coordinates and looking at homologous regions across species. For our pangenome pipeline, we started, for example, with the unaligned PacBio contigs, and then we used a variety of tools, pairwise approaches as a start, things like MiniProt and Minimap and DipCall, all by Heng Li, who is a part of this project. And MiniProt, for example, is basically a blast kind of approach to map proteins to genomes. And DipCall is a method for capturing so-called confident regions that are uncompromised by structural variants, particularly important for SNP calling and SNP analysis. We used a variety of tools that I think you've heard about in the workshop so far, like Minigraph and AGI and the Pangenome Pipeline by Eric and Andrea. And they've been really, really useful for this project. And then finally, we've used a bunch of visualization tools from AGI, including AGI Viz and Bandage. Now, this is just an example of our, it's a so-called PSMC plot. And what it shows is estimates of the demographic history of each of the 3 species, as well as one additional species, the California bird, using a method that basically looks at how long different runs of different levels of heterozygosity are across the genome. And it allows you to sort of look back in time and get an estimate for every individual in the study. And what this shows is simply that our hope that these birds would vary in population size turns out to be correct. So you can see the island birds in blue, very low heterozygosity, not much going on there at all. The Florida birds are sort of intermediate in green. And then finally in red, the Woodhouse's scrub jays have quite large population sizes. This is on the y-axis, we've got a measure of population size. So this is great. This was a big relief. It shows that our context of the study was appropriate. So the first thing that we looked at naturally were retro elements and repeats. And what's striking about these genomes is that these birds have over a quarter of their genome. In fact, this number has been updated. It's almost 30% of their genome is repeats. So you can see this is a lot bigger than what we typically captured from short read data. And so this is really exciting. And we think that most birds, their estimates of repeat abundance will have to be revised once we start using long read data. And just a look at some of the satellites. We're getting, satellites are turning out to be quite interesting in this group of birds. You can see a bunch of satellite families identified by repeat modeler, and how some of these families, encompass quite a bit of the genome. Here's one that takes up 30 megabases, for example. Some of these have unit repeat sizes of over 10 KB. And so this is a part of the genome that we simply don't see with short read data. We found one satellite, which was quite interesting. It was 18 kilobases of a unit. And this is just simply shows a variety of species on the x-axis including this mushroom, which has this really tall bar there. But I want you to focus on the two scrub jays, which are right here, and which have actually more satellite fraction of their genome than the human genome. The human genome is on the left, CHM13. Remarkably, these scrub jays have higher fraction of satellites than the human genome. And much of this you can see is these large satellites of greater than 10 kb unit size. So this is a big surprise, completely unexpected. And you can see how the frequency of this 18 kb repeat unit satellite varies drastically among the three species of birds we're looking at. We don't think this is actually an effect of necessarily population size. It's clearly some mutation that removed this satellite in the Florida and island birds. And what part of some of this variation is due to some satellites being on the sex chromosomes as well. So it's varying between males and females. But this is really quite a dramatic shift in the satellite landscape between these very closely related species. Now, telomeres are a type of satellite, and we've been able to look at these in really amazing detail. And of course, you're probably aware that telomeres shorten with age. And in this study by a group in Sweden, they showed that birds that were infected by malaria in the gray line here, their telomeres shortened faster than birds that were uninfected in the black line. So telomeres are quite of interest to ecologists that study birds as a measure of stress. And this is, you know, what a bird telomere is similar in many ways to a human telomere. Lots of these 6-mer repeats. And if we look at the length of the telomeres in these genomes, they appear to be somewhat longer than those in the human genome. So on the left, you see a distribution of lengths of human genome telomeres. And on the right, we have sort of a preliminary measure, simply sort of using blast tools of measures in different parts of the scrub jay genome. And so this is sort of work in progress, but we think that the repeats we're finding for telomeres tend to be a bit longer than what we see in humans. Now, we've been able to map these telomere sequences to these scaffolded chromosomes, and you can see that most of them do in fact end up on the ends, as we would expect. But we see a fair number of so-called interstitial telomeres, and these are not unusual. They've been seen in lots of different vertebrates, including humans. And they might indicate past chromosomal rearrangements. Now, we've been able to look at this shortening of telomeres over time by looking at the Florida birds. And so you can see here a measure of age on the x-axis, precisely known ages of birds, and total telomere length as measured by, these are actually queried using the satellite repeat finder developed by Hang Li on unassembled PacBio reads. And as expected, the line goes down. It's not quite significant. We actually are going to add a few more birds that are greater than 11 years old to this graph. These are going to be some hard-won data points. We're going to do some PacBio sequencing on 2 more birds, but we are hoping to get a significant result. But you can see that clearly, these telomeres are shortening over time, and they're it's just really neat to be able to measure them so precisely. Let's start looking at some individual regions, and we've used the AGI-Viz tools to look at different regions of different dynamic complexity. So here, for example, is a 400 kb region of the Hawks cluster, which is, of course, is very conserved. We expect to show very few structural variants. And indeed, we don't find that many. We see one here, which is about a 7 kb loop, which we can visualize with bandage here. And we also see a smaller one farther down. Now, we can think about a measure of complexity in these sort of linear representations called pangenome depth, the depth of a pangenome graph. I think you probably talked about this in your workshop, but just to review, a part of the visualization, which is either white or gray, These are regions of low graph depth, and they typically correspond to single copy regions or regions that are very conserved, or regions that don't have a lot of repeats in them. By contrast, you've got regions in red, which show are likely high complexity and repetitive regions. And you see those in this graph here. Most of this region is very conservative, but you do see these bright red sections indicating lots of structural variation going on. And we can also visualize this in typical tools like IGV, which is quite handy for getting a linear depiction of these structural variants. We've been able to find out where genes actually intersect with some of these structural variants. And we've been interested in whether there's actually a difference among these species in the copy number of these genes. And what this shows are the results of mini-prot, where we blasted the scrub-jay genomes with zebrafinch ref-seq sequences. And you can see that the number of genes across, these are only including the genes that vary among species. And you can see that the island species in the middle does seem to have a lower number of genes than the Western birds on the right or the Florida birds on the left. So there's some interesting dynamics going on. We can look at some of these examples in detail. So here, for example, is a gene called NPBWR2, which, again, this graph is colored by depth. You can see that most of it is quite low depth complexity, but we've got, a big insertion deletion polymorphism, in this case, covering part of the exon of this gene, and this gene seems to vary in copy number between the species. Here's another example of a different gene, which here shows that it's in a much more dynamic region. This again is colored by depth, so the red regions have lots of repeats in them. And you can see that the mini-prot hits at the top here are variable and likely have experienced a big insertion in the Florida birds, this gray region here. And on the right, you've got the bandage depictions as two-dimensional depictions. So interesting case of structural variants intersecting with genes. I'll say just a few words about the MHC. I think, am I doing okay for time? I probably have 5 more minutes or so. You can see the MHC has been of long interest to me, as the introduction indicated. It's a region that has a lot of interest in terms of molecular evolution, in terms of gene family evolution. It has direct links with disease and parasites, and so is of interest to a wide range of kinds of biologists. And here's just a depiction. These are not to scale. It turns out the chicken MHC is much, much simpler than the MHCs of humans and mice, for example. Initially, the chicken MHC was only found to be about 100 kb, whereas in mammals, it was much more like 4 or 5 megabases. The chicken value has been upwardly revised somewhat, but it's still much simpler and gene poor. And what we're learning basically is the chicken is not a very good representative for other birds in this case of this particular gene family. What we found at this stage, simply doing blasts, is that a typical haplotype of a scrub jay, this is mostly from the Western birds, will have about 5 or 6 of the so-called class 2 beta chain genes. These are canonical MHC genes. And we've used these, the gene in green and the gene in red, kind of as landmarks to look at the structural complexity of these regions. But what was a big surprise for me was just the complexity of the MHC class 2 region in these birds. You can see that basically every haplotype is distinct, has a different number and orientation of genes. And this sort of variation is just unprecedented. I actually did my postdoc using PCR to sequence and clone these genes, and we had no idea that this kind of complexity was lurking underneath once we look at with these high resolution tools. Just the number of hits. And one thing we found, interestingly, was we've been able to map the sequence differences of genes on individual haplotypes. So we can take the haplotypes from the PacBio assembly and ask, well, what's the diversity of genes on those haplotypes? And that's what you see in this plot, where the tree as a whole is all of the MHC diversity within the Western birds across individuals and haplotypes, whereas the red is the diversity of genes found on a single haplotype. And what's interesting here is that diversity across a single haplotype encompasses almost all the diversity across the whole species. And this might suggest sort of a new pattern of evolution, where the organization on haplotypes is designed to preserve a lot of diversity, because that's what the MHC is all about. Here's just some more examples of different haplotypes. Each tree in red indicates diversity on a different haplotype. And you can see that in almost every case, the haplotypes are preserving a lot of different diversity. So this is quite interesting and unusual. And when we look at the MHC in terms of its structural diversity, you can see it's quite lots and lots of indels. This is a plot that Eric actually put together for me, showing some of the complex repeat structures in the vicinity of MHC genes. There's a lot of satellite DNA going on. So this no doubt contributes to some of the diversity of gene number across these birds. We can look at the MHC region in terms of graph depth. And again, what we see are hotspots where we have these extremely high depth regions in red and purple, surrounding the more conservative regions in gray. And this underscores the fact that these MHC genes are surrounded by these haplotypes, by these satellites. And it likely is an important mechanism for them generating diversity. It's something we haven't really seen before, but which we think is actually present in other birds as well. And when we compare the MHC and the Hawks region, as expected, the MHC has a lot more structural diversity. That's what you see in orange here. And the repeats there are not only more numerous, but also longer than in the Hawks region. So this is all using the VCF tools generated by the pangenome, by PGGB. And then if we, just to end, we've lately been trying to look at the frequencies of structural variants across these three species. In many ways, this is the main goal of our study, to see how the dynamics of structural variants vary across these species of different affected population size. And the three species are up at the top. AC is the Florida bird, AI is the island bird, and AW is the Western bird. And what you can see is that these are structural variants of different lengths, and we also have SNPs up there in the upper left. The island birds do seem to have a higher frequency of so-called derived alleles, whether SNPs or structural variants. And these are, of course, alleles that have arisen since the divergence from the outgroup. And they allow us to look at the so-called unfolded site frequency spectrum, where we not just the frequency of an allele, but whether it's derived or not. And so this is still work in progress. You know, we see some peaks in the middle of these distributions, which could be spurious. They could represent simple gene duplications that we haven't accounted for. But this is the kind of data that we're really aiming for, and we're hoping to find evidence for an influence of population size on the distribution of structural variants. So these are just my conclusions. It's a very exciting time for pangenomes of birds and other organisms. And here's the team. We've had a lot of help from Andrea and Eric and his crew, and we've got lots of folks providing samples. And I'm just sort of the resident bird watcher who kind of watches all this amazing work go on. So I'd be happy to take any questions if you have them.
Thank you very much for this awesome talk, Scott. And I'm really happy to see that you had so much fun applying these most recent pangenomic methods and that you were able to get these awesome results with it. Are there some questions here in the audience? We need to allow Peter. Peter, could you please write your question in the question and answer section? That was a great talk, he says.
Thank you. I'm curious if in the workshop, are most people like human geneticists or microbial geneticists or?
No, here at the workshop it was a very mixed audience. And he's actually curious about how you called the SNPs.
Right, thank you. So what we've done so far in the case of SNPs is we tried to confine attention to these dip call confident regions. So these are regions identified by Hengley's tool dip call that are sort of free of large structural variants. We think this is perhaps a safer way to call SNPs or to confine our attention to those regions. You know, other than that, we have not done any sort of quality filtering. I mean, if you look at the quality of our contigs, they're all extremely high. I mean, they're all like 35 or 40x coverage. And we basically just used SNPs called from the PGGB VCF file, confining our attention to these confident regions. So that's what we've done so far. If you have any additional suggestions, it would be great to hear them.
Thanks. Any more questions?
Ditto about the car talk. Sorry. Second. I know Michael Hiller was looking forward to hearing your talk, Scott. My question, I've been traumatized by reading Michael Lynch reviews on selection versus drift, in particular the frailty article in PNAS and then the genome fart chapter in his book on genome architecture. And I noticed, so I came away from reading Lynch thinking that effective population size, from maybe my misreading of his work, is that you really almost have to be a bacterium for selection to work effectively. Now, I know that can't be possibly true, but I'm just wondering, in your work with the range of effective population size among the J's, do you really have enough dynamics there to tell whether it's selection or drift? I guess that you've answered that, so maybe answer it for a dummy.
No, I think it's a reasonable question. I might, you know, and right, I've drunk deep the Lynchian view of effective population size, and you know, I would say that I mean, we know that the effects of hitchhiking and linked selection do vary a lot in complex eukaryotes. I mean, we know that, for example, the level of neutral diversity that is actually achieved by a species that will vary a lot depending on whether it's a big population size or a small population size. And so I think it might require a slight update of Lynch's view, but my guess is that, you know, humans and species with even, you know, humans have relatively low diversity, but there are lots of species with higher diversity. But I think that the efficacy of selection will vary among those species. And, you know, whether or not these J's would show, I think, I actually haven't looked at the range of effective population size, but it's likely on the scale of 100-fold. And in other words, the Western bird is a lot more diversity than the island species. But yeah, that I think, so I'm cautiously optimistic. But I agree that estimating the fitness effects of structural variants is difficult. There is some software tailored to that. Some of Brian Charlesworth's graduates from his lab have some nice packages to estimate selection coefficients. And these, of course, are based on the frequency of variants. And so it's in some ways kind of circular. But we'll see what it tells us. I think I will say though that the island bird in our study, in fact, if anything, has a smaller genome than the other birds, and that of course is complete opposite of what Michael Lynch would predict. And so I think we're going to find some surprises for sure.
Yeah, he's very satisfied with your answer. Good job. Okay, so we take one last answer from the questions and answers section from Gabriel. Do the assemblies need to be perfectly curated following curation to be used as input for variation graph building? So for example, if we suspect that some context might be assigned to the wrong haplotype, would that be an issue? And will you do the DFE on these structure variations in different NE species? And he also compliments the talk.
Yeah, no, that's a great question. And, we're pretty much confining our inferences to what's going on individual haplotypes, on individual contexts. In other words, we can't say a lot about long range linkage disequilibrium, for example, because, we don't know, these haplotypes are numbered arbitrarily for any given region of the genome. And so haplotype 1 and 2 in a particular region of the genome won't necessarily be on the same chromosome as haplotype 1 and 2 farther down. And so all bets are off there. But within individual within individual contexts, some of which are quite long, we can do a lot of fun stuff that way. And so we're basically taking the Hi-Fi ASM output as a given in terms of the phasing. And we're pretty confident of that. I think with the Florida bird, in principle, we could actually test some of this because we have pedigrees, and we could actually look and see if markers are inherited in a linked fashion. But so I think given those constraints, we can do quite a lot with the phasing that Hi-Fi ASM gives us. Yep.
Thanks. And with this, we want to close Scott's talk, and let's thank him again, please.
Thank you so much. Thank you. Have a great workshop.
Thank you, Scott. Have a nice day.
Bye.
All right, we need to wait for Christina Boucher to sign in.
Hi, I am here.
Hi, Christina, we can hear you. Awesome.
Okay, now I can share my screen.
Yes, please. It works.
Yep, I'm just going to the slideshow. Okay, should I start?
Yeah, give me a time for a brief introduction, then you're good to go.
Okay.
So it's my pleasure to introduce Dr. Christina Boucher, Associate Professor at the Department of Computer and Information Science and Engineering at the University of Florida. Dr. Boucher started her career as a doctorate candidate at the David R. Sheridan School of Computer Science at the University of Waterloo. Then she continued her career as a NSERC postdoctoral fellowship at the University of San Diego under the advice of Pavel Pevsner. Her research focuses on the development of algorithms and data structures that enable large-scale sequence analysis. And today she will actually present her current advances of building pangenome indices for the sequence alignments against such pangenomic models. Christina, would it be fine for you if we actually record this session?
Yeah, absolutely. I think I sent confirmation of that.
Cool, great. Then it's a pleasure to have
email.
you here, and please start.
Okay, thank you very much. So as was mentioned, so today I'm going to talk about some of the algorithms behind, my slide advancement isn't working. Wait 1 sec. Still not working. Slide control. It's not.
Tab to your slides and say Zoom. You're in Zoom right now. With your slide.
I am on my side. Setup slide. Okay, that should work maybe. Okay, there we go. Okay, sorry about that. So as we sort of saw in the last talk, read alignment has been really important for a number of for understanding our knowledge of a number of different species. Here I've focused on the human genome. So we actually know that our knowledge of the human genome has been expanded by short read aligners in a number of different ways. So we are able to identify over 3,000 different structural variants just by looking at the 1,000 Genomes Project data alone. And I should say those 3,000 structural variants were identified only using short reads. And then if you look at even deeper coverage of these genomes, even more structural variants can be identified. But so today I'm going to talk about a bit about the algorithms behind the read aligners. So most short read aligners, including Bowtie and BWA and many other short read aligners, use something called Burrows-Wheeler transform, or the FM index, as we're going to see within this talk. And when you're looking at Burrows-Wheeler transform, or BWT, there's two specific operations that you need to focus on. So the first one is count, which given a specific pattern P, so this would be like our sequence read and our sequence data S, count is going to find the number of possible occurrences of a pattern P within S. And then locate is just going to return all the locations of each one of these occurrences. So it seems like this is really a simplification, but these two support of these two operations can be quite powerful. And in fact, this is the only thing that is used by methods such as BWA and Bowtie. So next, I'm going to explain a little bit about BWT. So you start with your input sequence. So this would be your FASTA file with respect to considering read alignment. And if you're considering more than one sequence in your FASTA file, then you would simply concatenate, or the methods concatenate all sequences together, separated by a delimitator symbol. So this is what our $ symbol is. Then we're going to consider all possible rotations of this sequence, and then we're going to sort these rotations in what is called lexicographical or alphabetical order. So this is called the BWT matrix M, which is commonly denoted as M. And one thing that you're going to notice is that the starting location of every single one of the rotations is stored along with the rotation itself. So why this is important is because of the lexicographical ordering, every single pattern is going to occur together within within a consecutive range from left to right, because they were sorted alphabetically. So for example, if you're looking for all A's, you can identify them looking at this looking for the contiguous range, and then all C's are going to occur together, and then even longer patterns such as AC are going to occur together within a contiguous range. So if you want to focus on count, for example, all you need to know is the size of this contiguous range. Now, one important aspect about a Burroughs-Wheeler transform is that it has this special property. Now, as you can see, M takes up quite a lot of space. So in practice, we wouldn't want to construct the entire BWT array. Instead, what methods such as BWA and Bowtie do is they store the first column, which is called F, and the last column, which is called L. And how you can support count and locate is because there exists a special property between L and F. It's called LF mapping. LF mapping states that the ith occurrence of a particular character C within L occurs, and the ith occurrence of that same character C within F correspond to the same occurrence in S. Okay, so that's why I've added this little number on this little numeric subscript on every single one of the characters in L and F. So that number corresponds to the number of occurrences that have been seen of that character prior to it. So using that value, you can actually identify the same character in L in the F column. Okay, so that seems a little bit mysterious, and you might be asking, like, where would you actually use this property and why would you use it? Well, using LF mapping, you can actually identify the same character, which allows you to search for any particular pattern. So here I'm going to show a very small example of this, but this gives you an idea of where you use LF mapping. So here I have my sequence S and my columns F and L. And in this small example, I'm not even using the DNA alphabet. So you can actually use any well-defined alphabet to construct BWT and the FM index. So here I have my matrix M and F and L, and then I take any pattern P, and I want to know how many times does P occur within S. So in order to do that, I'm actually going to process P from right to left. So I begin with my column F, and I find the number of times my rightmost character occurs within F. So that is going to be this consecutive range. And then I'm going to ask myself, well, where, if I want to go to the next rightmost character, which is equal to a B, how do I find that using F and L? Well, going from F to L is quite easy because every single one of these sequences is a rotation. So that's kind of the secret, is knowing that this represents a set of rotated sequences. So if I want to find all rightmost characters before this range, I just need to shift over and look at this corresponding range within L. So that's how I go from F to L. Then I can look at this consecutive range and ask, where are all the Bs? And that's easy to do. I can just look at the specific range where there exists Bs. So now I've matched A and B, but now I need to go back to my F column to recurse. So in order to accomplish that, what I actually do is use my LF mapping. So this is going to allow me to go back to my F column from my L column. And from there, I can just recurse and do the exact same character. So I would look at the next character, match that character, look at the same consecutive range within L, match the next character, and then use LF mapping again. So this is how you would find count. So count is just repeating this same operation until you've matched all the characters. And then the length of that consecutive range is going to correspond to the number of times that it occurs within S. Now, if you want to actually know the location of every single one of these occurrences, that's where you use your suffix array. So suffix array is the starting location of every single one of the rotations. So after you find your consecutive range, using that same little algorithm that I showed you, you would look at that same consecutive range within the suffix array, and that would be the locations of every single one of those operations. So the FM index, which is what BWA and Bowtie construct, is the Burrows-Wheeler transform, which is the F and L column, and the suffix array values. So together they correspond to the FM index. There exists some smaller auxiliary data structures floating around to support rank, which is the subscripts that I identified, but this is the basic data structure. And together they can support both count and locate. If you just want to support count, for example, then you don't need the suffix array values. Traditionally, you want to find all the locations of every single one of the values of all the occurrences. So that's where you would need the suffix array. Now, how BWA and Bowtie work is they use a seed and extend approach to find all alignments of all the sequence reads. So given a FASTA file, you're going to build an index using the FM index. And the FM index, one thing I want to note about it is that it's great for finding short exact matches, or I should say just exact matches, but it's not so good at finding mismatches, insertions, and deletions. So this is where the FM index kind of falls down. We say sometimes that it's blind to mismatches because it's unable to match any character. It's unable to find mismatches easily. So what Bowtie and BWA do is they find short exact matches between the sequence reads and the indexed sequences. And then they use dynamic programming or other chaining algorithms to fill in the alignments between these short exact matches, or what we call these seeds. The challenge with current methods is that the FM index requires on the order of a linear space in order to construct. So if you have around 6 terabytes of data, which is approximately the 1,000 Genomes Project data, then you would require a machine that has about 6 terabytes of memory in order to construct that. And that's unreasonable for most labs. So one of the goals that I've been working on and a number of other people have been working on is how do you build this index in sublinear amount of memory such that you can still support read alignment. Okay, so sublinear amount of memory would be using 1 terabyte of memory in order to index 6 terabytes of data. So even before short reads were even generated widely, people were working on this problem within data structures and algorithms. What they were asking is, can you support count and locate using a smaller data set, a smaller data structure? So in order to talk about that, we look at the last column, or what we say the BWT array in our matrix M. And what we're going to see is that it contains what we call runs of the same character. So Here we have a run of 3 C's, then $1 and then 4 A's. So if you had large repetitive text, what you would actually like, such as the 1,000 Genomes Project data, what you would expect is that you're going to have very few but long runs. Okay, so instead of having 3 C's here, we might have 1,000 C's. We call the number of runs equal to R. So R is kind of a famous variable within this space, because what we're actually interested in is, can you, instead of constructing an index that's linear in the total size of the input, can you construct it in N? So this was an open problem for some time, but recently there's been a lot of efforts in this direction. So putting some pieces together, we actually demonstrated that you can build an index which requires R space. So how you do that is you simply store the first suffix array value and the first character within the BWT for every single run. So for all R runs, you're going to store essentially 2 things, 1 suffix array value and 1 character. Again, you're going to need some small auxiliary data structures. There's a special data structure called phi, and phi actually allows you to retrieve the missing suffix array values. So this is referred to as the R index, and it was open for some time as to how do you actually construct the R index in efficient memory. So instead of just knowing about the existence and the possibility of supporting both count and locate with this smaller data structure, the next question is, can you actually build it in efficient space? So in order to do that, we came up with a simple algorithm called prefix-free parsing. So prefix-free parsing takes in your input string S. So remember, this is going to be all your sequences from your FASTA file. And then 2 integer parameters. One is W, and then the other one is P. And what you do is you build a representation of S of containing a dictionary D, and this is going to be a set of unique sequences, and a parse P. And the parse P essentially allows you to reconstruct S using sequences from D. So this is, and after you have D and P fully explain S. So once you have D and P, which are smaller than S itself, you can eliminate them. You can eliminate your original input S and just focus on storing D and P. And from D and P, you can reconstruct both your suffix array values and your BWT, or what we say, the run length encoded BWT. The run length encoded BWT is where you store 1 character for every single run in the BWT. So here I'm going to quickly go over how prefix-free parsing works, and it's actually quite simple. So you start with your input sequence S, and I have a small example here at the top. And because we're considering the sequences as being rotations, you're going to concatenate W copies of whatever delimiter symbol you're using onto the end of S. What this allows you to do is it then allows you to consider S as a rotation, as a circular sequence. The next thing you're going to do is you're going to do a rolling hash across S. So what a rolling hash is that it considers all W length subsequences. So in our example here, we would consider GA, then AT, then TT, then TA, and so forth. And you're going to calculate you would take all sequences that are 0 mod P. So the hash that we actually use is the Karp-Raven hash. Okay, so what this is going to end up with, if you take all 0 mod P W-length sequences, then you're going to end up with a smaller set of sequences, which we call trigger strings. And these trigger strings all have length W. And that should be obvious. The next thing that you're actually going to do is you're going to find the location of every single one of these trigger strings in your sequence S. So here I've highlighted them all. And from this, you have your full, you have a parse actually defined. So what we mean by a parse is all sequences that begin and end at a trigger string. Okay, so we're going to take all sequences that begin or end at a trigger string. And the first thing that we're going to do is we're going to take those sequences and put them into our dictionary D. And we're going to eliminate all non-unique ones. Okay, so what D is going to be, we call it a dictionary. For this reason, is going to contain all unique sequences that are going to be contained within this parse. The next thing that you're actually going to do is you're going to store a parse P, which is essentially the location of every single one of the sequences in D. So it's a map as how you are going to reconstruct S using the sequences from D. So for example, here we have the sequence S starts with the first occurrence with the first sequence that is within the dictionary. Then it continues to the second sequence that occurs within the dictionary and so forth. So you can see one idea is that why this actually works is for large repetitive input is because if you have a repetitive sequence, what you would expect is that D is not going to grow enormously, right? It's only going to contain the unique subsequences. And P is just going to be a set of integers. So this is what is referred to as prefix-free parsing. And why prefix-free parsing is important is that it's substantially smaller than our original input S. And we can construct the BWT and the suffix array from the dictionary and the parse. Okay, so originally people constructed the BWT and the SA from the sequence S. Now we're going to only construct the BWT and the SA from the dictionary and the parse, which makes it substantially more memory efficient. I'm just checking on the time here. So to give you a quick idea as to why this you can construct the BWT from this input. I've illustrated an example here where I take every single sequence which is in our dictionary, and I'm going to consider all possible suffixes of every single one of those sequences. Now, why am I doing that? That is because every single one of these suffixes is going to correspond to one rotation within our BWT. So if I can correctly place every single one of these suffixes in BWT order, and I can mathematically prove that, then I'm done. I have everything that I need in order to construct the BWT. However, if for some reason I'm unable to do that, then I can't construct the BWT. So this idea is essentially placing every single one of these suffixes in the correct order. So one of the interesting points about prefix-free parsing was that we are able to mathematically prove that by considering three specific cases. So the first case is simply when you have a sequence when you have a suffix that is unique only to one sequence in D. So here we have GAT, TAC, and it only occurs within one of the sequences in D. So this is our easy case because we actually don't need to consider P. We don't need to consider anything except that particular sequence in order to correctly place it in BWT order. So to repeat, I only need to consider the sequence in D in order to correctly place that. Now, one more challenging case is that if I have a sequence that is contained within this set, that is a suffix of more than one sequence in D. So for example, I have TAC, which is contained within the purple sequence, and TAC is also considered within the blue sequence. Now here is a more challenging case because I have to consider the ordering between these two possible rotations. So here you're going to have more than one rotation, and you have to decide which one is going to occur first. But in order to make that decision, what we do is we use our parse P. So because we have P is going to guide which occurrence is going to come first. So in our scenario here, we see that the blue one occurs before the purple. So that rotation is going to occur before the purple rotation. The last case, and you might have imagined this already, is that if you have a sequence in T that is a prefix of another sequence in T. So for example, if we have $GATTAC and it's a prefix of some other sequence that occurs within D. The reason why this is kind of the hardest case. So in this particular scenario, we wouldn't have a solution as to how you would correctly place these and have the algorithm be efficient at the same time. However, if we look at our definition of how we created the sequences in D, remember they have to begin and end at a trigger string. What this would imply is that you have a trigger string in the middle of one of the sequences in D, which we know is not allowed. So this eliminates prefix-free parsing, essentially eliminates this hard case. So this is how prefix-free parsing works, kind of in a high-level view. Now, it works very efficiently in practice. However, as I mentioned before, this is on haplotypes of chromosome 19. And what we see is that the dictionary remains quite small. And this is partially because we're not making a lot of additions to our dictionary. As you go from 200 to 400 haplotypes, you can see that the size of the dictionary doesn't grow very much. So this is very good. One of the downsides is that the parse actually grows kind of linearly. So this is kind of the downside. So one of the things that we actually considered doing is modifying this algorithm to do prefix-free parsing of the prefix-free parsing. So here I'll give you a very high-level view. So in prefix-free parsing, we take our input sequences and we build the dictionary and the parse. And then from the dictionary and the parse, we build the BWT and the SA. And in order to do that, we can eliminate the original input. So that was the great aspect about PFP. In recursive PFP, which is our new algorithm, we actually throw out the parse. So we keep the dictionary because the dictionary is quite small, and we build a dictionary and a parse from the parse of the original input. Hence the name recursive PFP. Now, the dictionary and the parse of the parse is actually substantially smaller. So after you have the dictionary and the parse of the parse and the original dictionary, the idea is then to construct the BWT and the essay without the original parse. So this is kind of the mathematically challenging aspect of the algorithm, is that now you're attempting on building the BWT and or the essay only using the original dictionary and the dictionary and parse of the parse. So in order to do that, we actually need to construct some auxiliary data structures. And then from those auxiliary data structures, build the BWT and or the essay. In practice, it does quite well. So this is how the construction of the BWT from the dictionary and the parse. And then this is the, in orange is then recursive PFP and the construction of the BWT from recursive PFP. And you can see that the memory usage drops substantially from constructing the BWT simply from the original dictionary and parse. And the running time actually remains more efficient. But really the goal here was the memory usage. The running time is just to ensure that we didn't substantially increase our running time. So the running time still remained low. But the memory is the important thing. That is what we really wanted to reduce. So just to recap, so prefix-free parsing is one way that you can actually build pangenomic indexes. And because it works very well in practice.
Christina?
Yeah.
So, sorry to interrupt, but we're a little bit running out of time. So if you could finish within the next 1 or 2 minutes.
This was my last slide.
Thanks.
Okay. Anyway, I'd just like to thank the students and collaborators on the project and our funding agencies. So are there any questions?
Thanks for the talk. Are there any questions here in the audience? Are there questions online? No. Okay. Then I will ask a question. So you presented a very general way of creating these indices, and I'm wondering if so in genome we sometimes have complex regions and non-complex regions, and if it would make sense to actually build an index specifically for complex or non-complex regions, for example, to be more efficient, or if the general solution is always the best way to go?
Do you mean like building a alignment index for complex regions, for example?
Sorry, again?
So I guess I can answer that question. So I think you can run, and my students are experimenting with this, as to how to change the performance due to the repetitiveness of the data. So if you're in like a repetitive region, then the values of W and P are going to have to be modified in order to gain the optimal performance. So my suggestion, so what we've been kind of working on is how does W and P actually modify the performance of the algorithm? And can you link that to the repetitiveness of the data, i.e. the value of R?
Okay, got it. Thanks. We have one more question from Eric Garrison. how many genomes do you think you can build an index from? Like, how large can you scale?
I think the largest that we had was from the entire 1,000 genomes project data. That was kind of like we tried it for all the data that we had publicly that we had access to. And we focused on the 1,000 genomes project data. And we actually looked at separating out the haplotypes. So in that one graph that I showed you, we looked at, I think, 2,400 haplotypes for full human genomes. I think that's the largest that we ran it on.
All right. And there's another question from Sarah Sherman. How intensive is it for the computer to generate these components, like the original dictionary, the dictionary, and parse of the parse from the file S? Or in other words, is it intensive to break down the sequences into a format where they can be reconstructed to derive BWT and the suffix array?
Yeah, that's a good question. Actually, I was going to try to refer back to my slides. Can you still see them, or is that?
No, you will have to reshare, please.
Okay. So if you look back on this one slide here, what you can see is that the main focus was actually the parse. So the parse kind of grows a lot more closer to linear than the dictionary. The dictionary also always remains kind of substantially small, but the parse is what was problematic. Prefix-free parsing in practice works quite efficiently. I think that's why it's become like kind of popular, and I know that some people are including it in algorithms textbooks because in practice it's very efficient. And you can also parallelize it. It works extremely efficiently. Constructing the BWT in the suffix array from the output of prefix-free parsing is a lot more challenging. You have to consider all possible rotations of an input string, and then you have to attempt on correctly placing them. And it's not as easy to say parallelize that. But there's a lot of work in this area by myself and many other people.
Thanks.
So I would say that's kind of an open problem as to how to reduce the current, how to reduce the construction.
I have one last question. So we also build pangenome graphs, and I'm wondering if you could also index these, or what's the current status and research there?
Prefix-free parsing to construct a graph from the PFP. Yeah, we've thought about it. And I think that there are some ideas like floating around. I think I've talked to Nicola Pretta about it, and who's in Italy. He was one of the founders of the R index. He's more theoretical. And also Travis Gadji is also interested in that problem. So there's a lot of ideas floating around, but there's nothing published at the moment. So it's still kind of like open. I guess the main issue as to why is because people like Nicola are interested in, can you store a graph in R space? And that's kind of the first natural question that you would have. And then the next question is, after you define the data structure, how do you build the data structure? So that's kind of like the natural order of how you'd answer the questions. And we don't have a good representation for, we don't have an R index representation of graphs at the moment.
All right, so we're looking forward to a solution.
Yeah.
I think that's it. Let's thank Christina again.
Thank you.
Yeah, thanks for the talk and have a nice day. And please have lunch and have fun. So we'll start here again at 2. You have around 1.5 hours and you have time to freshen up.
Okay, welcome back, everyone. It's my pleasure to introduce Dr. Joop de Legit. Joop did his PhD in bioinformatics in Netherlands on pediatric diagnosis. Then he did 2 postdocs in cancer genetics and personalized medicine, after which he moved to New Zealand as the senior science lead bioinformatics and genomics. His team uses genome sequences, important understanding and improving health, and can be used to to define child disorders. It's also used to investigate foodborne outbreaks and helps understand and combat the rise of drug-resistant bacteria. Dr. Joop spent most of his time helping to support and guide SARS-CoV-2 analysis in New Zealand. Today, he will explain to use the pangenome graphs in pathogen genomics for public health. So it's our pleasure to have you here, Joop, and you can start your talk.
Thank you, Flavia. And thank you for having me. Yes, so unlike my accent might suggest, I currently live in New Zealand. And I would like to open with a formal Te Reo Māori welcome, which is of the indigenous population of New Zealand. Tēnā koutou katoa. E mihi ana ki tēnei kaupapa a toto, he tangata tiriti aho. Kei te mahi o ki Esarpo whakahere o ropu genomics and bioinformatics. Ko Joop de Legit taku ingoa. Tēnā koutou, tēnā koutou, tēnā koutou katoa. That's just a welcome to everyone here and also online. But it's also a recognition of the work that was done, because a lot of the things that I'll be presenting is only possible because of the work that went into PGGB and in the VG and the pangenome field in its own. And we're, in the end of it, we're more the end users. We try to apply it. So in terms of I'm presenting in the US, and I did have to fly an awful long way here, and Nanopore provided some sponsoring because I was also speaking at their conference in London calling. But they didn't pay for me to be here or have any influence on the slides whatsoever. And I have some conflicts of interest within New Zealand, where I'm also an associate at Otago University. So getting that out of the way, the institute where I work, we do a bunch of different things. It's all about people in the end of the day, and it's about applying that in sometimes a clinical or environmental setting. So we've used genomics to determine whether a tusk was actually of a woolly mammoth or of an irregular elephant when it came across our borders. We've used it to study which animals are contaminating our waterways. We've used it to look at different plants, and if you put those on fields, if they actually help with the nitrogen cycle. We've looked at food safety, as was mentioned by Flavia, in terms of if there's a outbreak, is there a food association? Is there a particular product that we need to recall? And also some human genomics, but I'll talk mostly about public health. So most of you don't need to hear this, but reference genomes, they're useful, but they're not ideal. Especially if there's large pieces of the DNA missing. So for humans that are of diverse ancestry with the previous genome, there might be over 10% of your data that's missing. But that's even worse for bacteria. So the people that were in the course, we had a bit of a discussion about closed and open and whether that's actually a binary thing or not. But what I can definitely say is that humans exhibit a lot more closed genomes. And part of that is because they sexually reproduce. Bacteria, they're a lot more promiscuous with their genomes. They can suck up DNA from their environment or from nearby sources. So they exhibit a much more open genome. So actually, the sort of the concept of pangenomes came from that field, and we're hoping to now contribute it back to it in a functional form. And one of the things that I also want to point out is that most of the work that I'll be presenting was actually done by Zoe Young, who's a postdoc in my group, who had a lot of help from Andrea and others to do all of this work. But basically what we're showing here is that in bacteria, there's a part of the genome that's basically present in all bacteria, but everything else is basically almost species specific. So it's really a problem where if you try to find a single reference, it will always miss things just by the nature of how open those genomes are. So what we figured is that now that pangenome graphs are becoming more of a thing and that the tools are coming to a utility stage, we could start leveraging that to not only look at the core genome, but to start looking at the variation that's present in the pangenome. Because especially as you were dealing with an outbreak situation, every single SNP matters because it might decide treatment options or it might decide a recall of a food product. So you better want to be sure and you want as much resolution as possible. So what we are thinking is that, well, if you have those pangenomes and you have those graph representations of those, that you can study those variations. So that, for example, in the case here, there's the purple ST42, that's a sequence type. You could study this variation if you have that graph, and you can study it within that particular sequence type. Whereas if you would have the normal reference, that information would be missing. You wouldn't even be mapping it. You can't even study it. So that's what we did, and we've used PGB for most of that. This is the pipeline that we have. The slides will be available, so I won't go into too much detail, but there's two parts that I will focus on that are especially relevant in public health, and that's the distance that you can compute along your paths to actually do phylogenies, and it's genotyping and variant calling. Now, most of the work that I'll be presenting was done on an epidemic outbreak of Neisseria meningitidis. For those of you that don't know that, it is a gram-negative bacteria, and it causes meningococcal disease. And then you go, well, what is that? Well, it actually causes, in severe cases, severe swelling of the tissue surrounding the brain and spinal cord and can lead to death. And in between 1990 and 2008, New Zealand had a prolonged epidemic of a particular Neisseria meningitidis that we refer to as New Zealand mengen B. So that's the abbreviation that I'll be using. And then vaccinations actually meant that we got that epidemic under control and are now sort of at more normal levels of this particular bacterium. And there's three major sequence types, or clades they're also referred to. So that's what you see here in the top hand corner. They're quite genetically diverse. And there's three major rearrangements that define these. So what I've shown here is a mauve representation of those genomes, and then on the bottom there's the 2D genome graph where the colors actually represent the orientation of the genome. So black is forward and red is reverse. So we wanted to start with a benchmarking because I'm a paranoid person, so I want to benchmark before I start using things in anger. So I'll talk a bit about how we did that for both large structural variants and indels, and then talk about how we did it for phylogeny. And I'll talk a bit about whether we use short reads and if that's possible as well. So first of all, we wanted to create that ground truth set through simulation because I don't trust any single variant caller. So I'm going to put exactly in there what I want there to be in there, and then we can see what we find back. So we try to reflect the number of mutations that you would normally see, numbers of indels, and the large inversion events that we knew that were present. So now we have a ground truth set. And that ground truth set we can mutate further so that we can actually have a phylogeny. This is not sort of the real life phylogeny that you would get in biology, but at least it allows us to computationally assess how well the phylogeny is reconstructed and how it behaves in terms in the graph. So this yielded 120 genomes in total. And we used WGSIM to do that, and then also simulate sequencing reads based on top of that so that you can see sort of the performance if you would have a new sample. The ground truth comparison is pretty good. We missed a literal handful of variants, and that's just due to the ambiguity of calling indels. So these are the three examples of ones that we've missed. And that's just due to the fact that, as you can see the figure D there, there's a GTT-CGAT deletion, and whether you anchor that on the C or if you anchor that on the G that flanks it, that has to do with the variant call format. So we were quite happy with the performance there, that we found everything that we were expecting to find. And also in terms of the overall structure of the graph, in terms of the inversions that we put in there, all those regions were appropriately recapitulated and the breakpoints were exactly at those positions where we expected them. So for both SNPs, indels, and structural variants, we were happy campers. And then one of the big things that, of course, in bacteria with the reference bias, because no single reference can contain all of the genes, is mapping rate, because anything that's not in the map reference is not going to map. So here we have the simulated genomes, and we've compared those against their sort of closest reference that is publicly available. And then you can see that you get somewhere between 994 or 100% mapping. But you can actually see that if we use the graph approach, we always map 100%. And if we look at the number of high quality variants that we get based on the QC metrics, we can also see that if we map those against a graph, we get a higher proportion of high quality variants, again, because there's more support for those reads that align to the other regions. And also because there's the variant calling itself can learn from the other variants that we've seen previously. And we also simulated some more sort of real life reads that we can then map against it to sort of see how that influences matters, because of course, sequencing technologies are not perfect. None of them are. And probably never will be. So what you see on the left-hand side is those reads where we've sort of introduced errors as they typically occur in a sequencing experiment. And on the right, we have those same, but then on a clean basis. And this is again looking at the mapping rates. And what we can see is that introducing those mapping or sequencing errors has a very minimal effect on when we compare to the graph. So those are the bars in blue. Well, it has a larger effect if we do it against a single reference. Again, not unexpected, but these are the types of things that we want to be able to show people that you can really do these things reliably and well. So then we started applying it to some real data. So one of the first things we did was looking at particular drug resistance. And you can also already get some beautiful figures out, but you can also start to look at some of the phylogenies and sort of what they result in terms of the transmission of these elements. So we then took 130 public Neisseria genomes and looked at those structures. And if you remember from the previous slide, we had sort of those three major archetypes with those major rearrangements in there. And each of those is represented here. And if I can make one feature request, harder than horizontal sorting, I would also love vertical sorting of the 2D graphs. Because there's actually the three sequence types are quite well represented there. And also in terms of genes that are actually in that extra pangenome that we can now assess, that can be quite important genes. So this is my favorite slide. So we then looked at how well we can do the phylogeny in a reference-free basis. So on the left-hand side, figure C is the PGGB version of the similarity matrix that was computed. It took a day to compute, but it is lovely. For those of you that are not used to phylogenetic trees, that's the type of tree that you might want to take home. The other one is not so much. And what the biggest difference is actually that apart from the overarching structure, so the colors are supposed to cluster together. And you can see that they beautifully do that, but that actually also on the lower level, there's a lot of resolution. And that is because within those regions that are specific to that part of the tree, it can still detect the other variation. Well, if you look at the Kamer approach, which is sort of the next best reference-free approach, you do get that sort of overall arching structure, but you don't get that resolution that is much more fine-grained. And especially for outbreak detections and those types of things, that resolution really is a game changer for us to be able to detect the transmission order or pathways that these bugs take. So that is what we've done for Neisseria, and we're hoping to be able to do some more association studies on virulence and other factors. And now that we can also reliably assess that accessory genome. But some of the other things that we've been having fun with is the nanopore technology, because apart from accuracy, one of the really big things that we focus on is real time as well, because it's especially about the sort of clinical implementations. And we've been able to work with a conservation group in New Zealand that are working with highly endangered birds. So there's only about 250 of those birds left on the planet. And they actually can now use sequencing to sex these birds because they found that they were trying to mate two males and they weren't getting much success there. Because it's currently quite difficult to do that reliably, and they can now use sequencing on the island where they are. It's a sanctuary island. We've seen people over here in the US, Devin Drown, that has set up his master's students with a sequencing, and within a week they go from a master's student with some pipetting skills to be able to design and run their own genomics experiments. And also on the clinical side, the people are using it for tumor classification while the operation is ongoing, or to detect outbreaks on wards in real time and actually preventing ward closures or spread within those wards. So what I want to end up with is pangenomes love long reads, and they don't care if they're PacBio or ONT. It's just that I have experience with ONT that I can talk about that. Methylation is something that we're all going to see a lot more of, especially for phasing. I think it's going to make a really big difference because homozygous regions, you cannot phase with just genotypes, so you will need something else. I think methylation is going to be a big game changer there. So one of the things we did is a comparison with an Illumina array, and then do the same experiment with adaptive sampling on nanopore, and you can actually ascertain the same CpG size and get really good correlation there. And the other thing I think is that we've seen is then the last sort of year and a half is that there's a lot more sequencing companies coming out with some of the patents being broken up. And what is really exciting to me is that a lot of those are actually in the lower end in terms of getting started with them. So it's more accessible to third world incomes, and also more accessible to people that might be in resource constrained regions. So what I've presented has been funded by Genomics Aotearoa. So that's a large consortium in New Zealand that thinks about genomics. My own institute and the Health Research Council, and it's a collaboration with the University of Otago, Tennessee, and Massey University in New Zealand. And these are the people that did all the actual work. Thank you.
Thank you, Joop. So let's see, there are some questions in the audience. No, let's see in the chat. Otherwise, I have some questions. There is
My second or third? This one?
After that.
This one?
No, I like the 4 different what do we call this? Outcomes. No. You were at it before the
Okay. Yeah. This one?
No, the one with the 4 outcomes at the bottom.
Sorry.
There.
Yes.
Yes. So this is one of the most, at least to me, most simplified but yet explanatory slides during the conference. So we can see what happens with pangenomics, with genomics, and what you do. I like how it's tying to public health forensics, food safety, water, and the environment, and it's not. And it includes analysis and research. So I'm just saying thank you for that.
Most welcome. Thank you.
Thank you. I see another question in the chat, maybe. Okay. Do you think pangenomes could eventually be used to predict viral evolution pathways?
Yeah, no, I think that for viral species, there's still a bit of work to do for us to detect variants that are still arising. So if you're sequencing a virus, you're typically sequencing a large population of viruses, and a part of them will already have mutated and part of them will have not. It's a bit like cancer with the somatic variants. And actually, if you then in a second sample see that variant again, but now it's at 100% frequency, you actually want to go back to that original sample and see what the frequency was to see if it was already sort of, breeding, if you will. But by looking at these things in a conjoined fashion where you can look at the structural event as well as the SNPs, you will get a more detailed view of directionality. And I think we will get a better understanding of their dynamics as well if we can do this in these larger populations and sort of see evolution happening in real time.
Okay, great. I have another question. You are going into the pangenome? And are you able with the nanopore to solve also complex regions? So there are some complex regions that are you not able?
Yeah, so at the moment we've stuck with one technology. We know that there's certain regions that we will have a slight bias to, but it's already a lot less biased than with short reads. And for these bacteria, we can sequence quite deeply. So that sort of drop of coverage, we will still have 20x coverage, because the rest of the genome we typically have at like 300x. Because they're lot smaller genomes, so we're not as worried about sort of missing those sort of last bits and pieces as some of the people that work with larger genomes have to be.
Yeah. Okay, great. So I don't know if there are other questions.
Great talk. Using methylation for phasing is a very interesting idea. Can you talk more about that, please?
Yeah, sure. So there's some very great preliminary work in cancers where they've actually used it to detect those reads that are derived from the cancer and not from the environmental cells. And then can use that to phase the cancer alleles. And there's also people that are now starting to work on software where the different, if there's a stretch of homozygous material where you can look if there's basically a bimodal distribution of methylation, where you can then split out those alleles and you can phase over that homozygous region. And I think that as we are able to detect more types of base modifications, that will become more and more powerful so that we can actually separate out those alleles. And that's going to be especially relevant in those higher than diploid types of organisms where that phasing problem is quite wicked.
Okay, great. I have another question. So what's the next? I mean, are you going to study another similar organism, and will you expect the same complexity? Or?
Yeah, so we've sort of chosen Neisseria because as far as bacteria go, it's sort of middle of the road. It has a decent sized genome and it is promiscuous. It does sometimes take up material from the environment, but there are bacteria that are way better at taking up stuff from the environment. So there's definitely more complex ones out there. What the first thing that we want to do is sort of bring this now into a practical utility phase. So for the organisms that we work on as from a public health perspective, create these pan-genomes and put them into sort of a production setting where you can immediately ask the question, like, is this part of an outbreak? Because those phylogenetic tools are already there, but I think that you can actually leverage the structure of the pan-genome to do that more quickly. And that's sort of the real-time part of what we care about, is that if we can get an answer the same day, that could actually save lives. Because that recall might happen a day sooner.
It's a great way. So, okay.
Hi, thanks for the talk. I'm just curious about the mechanisms of virulence that you mentioned that you're starting to uncover now. Do you think these are more associated with the accessory genomes that you're able to detect with these open genomes?
So we haven't found anything just yet, but we're starting to look for it. But what we are now able to do is to systematically incorporate the accessory genome in a way that is uniform across the whole collection, because there have been some people that have done that, but it takes a whole lot of work to do that. So it's done for specific species for specific scenarios. What we're hoping is that this could be sort of like, a graph WAS, if you will, where you can do genome-wide association studies, but includes those pieces of DNA that are unique to a particular bacteria. And there's a lot more technological work happening in that space that we're hoping to be able to leverage and build on.
So maybe the last one.
Yep, can you just say something about the difficulty of the DNA prep in the field versus the fussiness of the platform?
Yeah, sure. So we do quite a bit of training and outreach because one part is that we really want to empower the indigenous people to also be able to do their own experiments. So we actually have had a group of people that were sort of lab trained that come in and use the technology. For us, what works really well is that with the nanopore, we can fill often and quickly. So if the experiment hasn't quite worked, if after half an hour of sequencing, we can look at the run and we can actually see that it's not doing, and then we can stop it and restart. If you need to wait a day for your sequencer to finish, that for training purposes, that's really hard. The other thing is that the rangers that I was talking about that are using it for these birds, they're doing this on an island where there's one flight a week, and that's like a little dingy aeroplane where you can, if you're lucky, fit 4 people, right? So they're very happy that they can run it there with a little solar panel, and that the compute is quite minimal, and that they can just have it sitting there on their bench, and they can actually do those experiments themselves. No. Now, if you really want to do ultra long stuff, I would definitely recommend doing that in a well spec'd out lab where you can control all the things. As long as you're sort of in the 20 kb range, you can do that in the field just fine, as long as you have good material.
So yeah, thank you for coming in person. And I think that we can go ahead with the next speaker.
But you really don't need to record this. But like what we want to point out, we confidentially call the loop here, we call it the Graffiocavallo. And that's like a tribute to the Caciocavallo, which is a cheese from South Italy. You can clearly see why there's a clear similarity. So I just want to provide, I just want to say a big thank you to all the instructors and helpers. You've been just great, and especially the junior ones, because they were great. So and I really loved how the dynamic was the structure, that people was a student and then was an instructor and then was a helper, just really a dynamic group of people that exchange knowledge. Besides that, the catalyzers of the event, I couldn't find any other word to describe that. Piotr is that's the reason why we're all here. Rob and Bob, which, provide major support for everyone, and Eric, and also Pedro, because Pedro actually started this tradition of the pangenomic course, and he's also, he got a lot of experience in training, and he's kind of teaching us also how to do training, and also a good babysitter, as you can see. And thank you to Melanie and Tamara. They were actually precious and and also patient. I just want to apologize for how, the complete lack of discipline in the rest of the group. Even with that, they were able to help us. And, so big thank you to Tamara and Melanie. And just a little story of how it all started. I was thinking about, I got a bit sentimental here. So it was August 2020, and Flavia was a master student in my lab. And so she came visiting, and she brought Andrea. And I can't remember why Simon was there, but just Simon came somehow. I can't remember why, but just yeah. So we all met in Italy, in South Italy. And I know we had just a lot of fun coding and eating and coding. And I was pretty much, we spent, I guess, 1 week together. I don't know how long it was, but it was 2020. And also exercising occasionally. And Simon still remember running in South Italy with 30 degree, at 2 p.m. in the afternoon. And then coding. And so just to say, it's a lot of work, which actually went through now 3 years almost. And I mean, I'm really proud to see how this, all this work actually, it ended up in beautiful things. And it actually ended up having all of us here. This is the very first day we came to Memphis of work. It was really nice. And we were planning to go for coffee, but the coffee place was closed. So luckily nearby, there's a High Cotton brewery, so we could go for a beer. And that was great. And yes, and so, big things are going on here, and also interesting collaboration. This is Piotr with people from Cornell. And how we'll continue. I think it's just this is just the beginning of how it actually will continue. And this is, just to say thank you to all of you who came here, because that make possible to build connection and to expand actually our range of possible collaboration. And this is it. And that's for people in the, yeah, that's Andrea's meme, adios. Okay, now this is it. Just a big thank you to everyone and see you next year.